What is the primary function of a microscope in physiology experiments? A. To measure blood pressure. B. To count cells. C. To monitor heart rate. D. To estimate hemoglobin levels. The correct answer is B. Microscopes are used in physiology experiments to visualize and count cells. Which of the following techniques is commonly used to analyze blood samples in physiology experiments? A. Electrocardiogram. B. Cell counting. C. MRI scan. D. X-ray. The correct answer is B. Cell counting is a common technique used to analyze blood samples in physiology experiments. What does hemoglobin estimation measure in the blood? A. Enzyme levels. B. Glucose levels. C. Oxygen carrying capacity. D. Blood pressure. The correct answer is C. Hemoglobin estimation measures the oxygen carrying capacity of the blood. Which of the following is an essential parameter often measured in blood pressure estimation? A. Heart rate. B. Blood glucose level. C. Respiratory rate. D. Pulse oximetry. The correct answer is A. Heart rate is an essential parameter often measured in blood pressure estimation. In physiology experiments, what does blood pressure measurement help in assessing? A. Hemoglobin levels. B. Cell viability. C. Cardiovascular health. D. Lung function. The correct answer is C. Blood pressure measurement helps in assessing cardiovascular health in physiology experiments. What is the main benefit of heart rate monitoring during physiological studies? A. To estimate blood volume. B. To assess cardiac function. C. To measure bone density. D. To analyze kidney function. The correct answer is B. Heart rate monitoring helps in assessing cardiac function during physiological studies. Which of the following is not typically measured in a blood analysis in physiology experiments? A. White blood cell count. B. Liver enzyme levels. C. Urine protein concentration. D. Platelet count. The correct answer is C. Urine protein concentration is not typically measured in a blood analysis during physiology experiments. What is the purpose of cell counting in physiological studies? A. To estimate blood pressure. B. To assess lung capacity. C. To determine cell population. D. To monitor electrolyte levels. The correct answer is C. Cell counting is used to determine cell population in physiological studies. What can be inferred from a high white blood cell count in a blood analysis during a physiology experiment? A. High blood pressure. B. Infection or inflammation. C. Low hemoglobin levels. D. Impaired kidney function. The correct answer is B. A high white blood cell count in a blood analysis indicates possible infection or inflammation in physiology experiments. What is the significance of platelet count measurement in physiology experiments? To evaluate liver function. To assess blood clotting ability. To estimate lung capacity. To monitor electrolyte balance. The correct answer is B. Platelet count measurement is important for assessing blood clotting ability in physiology experiments. In physiology experiments, what is the main purpose of using a spectrophotometer? A. To measure the absorbance of light by samples. B. To count the number of cells in a sample. C. To determine the blood pressure of an individual. D. To monitor heart rate changes over time. The correct answer is A. A spectrophotometer is commonly used to measure the absorbance of light by samples, helping in various physiological research and experiments. Which of the following devices is commonly used to determine the oxygen saturation in blood samples during physiological studies? A. Pipette. B. Spirometer. C. Pulse oximeter. D. Centrifuge. The correct answer is C. A pulse oximeter is a device frequently used to measure oxygen saturation in the blood, particularly effective in physiological studies. What does a centrifuge help in achieving during blood analysis in physiology experiments? A. Separating blood components based on density. B. Counting the total number of platelets in the bloodstream. C. Estimating the hemoglobin concentration. D. Analyzing the blood pH levels. The correct answer is A. A centrifuge is utilized to separate blood components based on their density, which is vital in various aspects of blood analysis and physiology experiments. In physiology research, what is the primary purpose of using a hemocytometer for cell counting? A. To determine the concentration of cells in a blood sample. B. To measure the absorbance of light by cells. C. To monitor changes in blood pressure. D. To estimate the oxygen saturation in blood. The correct answer is A. A hemocytometer is specifically designed to determine the concentration of cells in a blood sample, aiding in cell counting during physiological studies. Which of the following is a key advantage of using flow cytometry in cell analysis during physiology experiments? A. Measuring blood pressure accurately. B. Counting the total number of platelets in the blood. C. Analyzing multiple cellular parameters simultaneously. D. Determining the heart rate of an individual. The correct answer is C. Flow cytometry is advantageous in physiological studies for analyzing multiple cellular parameters simultaneously, providing comprehensive insights into cell characteristics. What information can be derived from a differential white cell count in a blood analysis during physiology studies? A. The total number of red blood cells in the sample. B. The proportion of different types of white blood cells present. C. The average size of platelets in the bloodstream. D. The hemoglobin concentration in the blood. The correct answer is B. A differential white cell count in blood analysis helps in determining the proportion of different types of white blood cells present, offering valuable diagnostic insights. Why is it important to monitor endothelial function in studies involving blood pressure regulation in physiology research? A. To estimate the oxygen saturation in blood samples. B. To assess the overall cardiac output of an individual. C. To evaluate the health of blood vessel linings. D. To determine the total number of platelets in the bloodstream. The correct answer is C. Monitoring endothelial function is crucial in physiology research to evaluate the health of blood vessel linings, providing essential information in blood pressure regulation studies. During blood analysis in physiology experiments, what does the erythrocyte sedimentation rate, ESR, indicate about the blood sample? A. The total number of platelets present in the sample. B. The rate at which red blood cells settle in a specific period. C. The amount of oxygen saturation in the blood. D. The level of hemoglobin in the bloodstream. The correct answer is B. The erythrocyte sedimentation rate, or ESR, reflects the rate at which red blood cells settle in a specific period. This indicates potential inflammatory or pathological conditions during blood analysis in physiology experiments. Which parameter is commonly assessed through a spirometry test in respiratory physiology studies? A. Heart rate. B. Lung volume and airflow. C. Platelet count. D. Blood pressure. The correct answer is B. Spirometry tests are frequently used in respiratory physiology studies to assess lung volume and airflow, aiding in diagnosing respiratory conditions and monitoring lung function. What is the significance of troponin measurement in physiological studies? especially related to cardiac function assessments? A. To estimate the oxygen saturation in the blood. B. To determine the number of platelets in a blood sample. C. As a biomarker for myocardial damage. D. To count the total number of white blood cells in circulation. The correct answer is C. Troponin measurement is crucial in physiological studies, particularly for cardiac function assessments, as it serves as a biomarker for myocardial damage, aiding in diagnosing heart conditions and assessing cardiovascular health. What function does a hemocytometer serve in cell counting during physiological studies? A. Measuring oxygen saturation. B. Determining cell size. C. Quantifying cell concentration. D. Analyzing enzyme activity. The correct answer is C. A hemocytometer is a specialized counting chamber designed to quantify the concentration of cells in a sample. It allows researchers to determine the exact number of cells present for various types of cells. Which of the following is a common application of a centrifuge in blood analysis during physiological experiments? A. Determining enzyme levels. B. Separating blood components. C. Measuring oxygen levels. D. Assessing cell membrane integrity. The correct answer is B. A centrifuge is used to separate blood components based on their density, allowing researchers to isolate specific components like plasma, platelets, and red blood cells for further analysis. What is an important aspect of blood pressure measurement in physiology experiments? A. Assessing heart rate. B. Monitoring cholesterol levels. C. Evaluating vascular function. D. Measuring brain activity. The correct answer is C. Blood pressure measurement helps in assessing the function of blood vessels, including their elasticity and responsiveness, which are crucial for maintaining proper blood flow and pressure in the body. How does a spectrophotometer aid in hemoglobin estimation during blood analysis in physiology experiments? A. Determining cell morphology. B. Measuring cell volume. C. Quantifying hemoglobin concentration. D. Analyzing DNA content. The correct answer is C. A spectrophotometer is used to measure the absorption of light by hemoglobin in a blood sample, allowing for the quantification of hemoglobin concentration, which is vital for assessing oxygen carrying capacity. In blood analysis during physiology experiments, what information can be obtained from a white blood cell differential count? A. Total leukocyte count. B. Composition of different white blood cell types. C. Hemoglobin concentration. D. Platelet function assessment. The correct answer is B. A white blood cell differential count provides information on the specific types of white blood cells present in the sample, which can indicate the body's immune response and potential infections or disorders. What is a primary advantage of using flow cytometry for cell analysis in physiology experiments? A. Quantifying enzyme levels. B. Determining protein structure. C. Measuring cell size. D. Analyzing multiple characteristics simultaneously. The correct answer is D. Flow cytometry allows for the simultaneous analysis of multiple characteristics of individual cells, such as size, granularity, and protein expression, providing detailed information for cell identification and classification. Why is monitoring endothelial function essential in physiology studies related to blood pressure regulation? A. To assess liver function. B. To understand muscle contraction. C. To evaluate vascular health. D. To measure lung capacity. The correct answer is C. Endothelial function is crucial for maintaining the health and function of blood vessels, including regulating blood pressure. Monitoring endothelial function helps in assessing the integrity and responsiveness of blood vessel walls in regulating blood flow and pressure. What parameter is commonly assessed through a spirometry test in respiratory physiology studies? A. Pulse oximetry. B. Hemoglobin levels. C. Forced expiratory volume. D. Glucose concentration. The correct answer is C. Spirometry is used to measure respiratory parameters, including forced expiratory volume, which helps assess lung function and diagnose respiratory conditions like asthma and chronic obstructive pulmonary disease, COPD. Why is cell counting important in physiological studies involving microscopy? A. To estimate the number of different cell types present. B. To determine the size of the cells. C. To identify specific organelles within the cells. D. To measure the cell membrane potential. The correct answer is A. Cell counting helps in estimating the number of different cell types present, providing valuable information for various physiological analyses. What does a buffy coat layer indicate in a blood sample during physiological studies? A. High platelet count. B. Elevated leukocyte count. C. Decreased hematocrit. D. Low hemoglobin levels. The correct answer is B. The buffy coat layer usually consists of leukocytes, so it indicates an elevated leukocyte count in the blood sample. Why is it crucial to monitor blood pressure continuously during physiological experiments? A. To assess the heart rate variability. B. To detect any sudden changes in cardiovascular function. C. To measure the oxygen saturation levels. D. To estimate the total blood volume. The correct answer is B. Continuous blood pressure monitoring helps in detecting any sudden changes in cardiovascular function, which is essential for understanding physiological responses. In blood analysis, what does the mean corpuscular volume MCV indicate about the red blood cells? A. Average size of red blood cells. B. Total hemoglobin content per cell. C. Oxygen carrying capacity of the cells. D. Red blood cell distribution width. The correct answer is A. The mean corpuscular volume, or MCV, indicates the average size of red blood cells. It provides information about their morphology and potential health conditions. What does the pulse oximeter primarily measure in physiological studies involving oxygenation monitoring? A. Blood pressure. B. Heart rate. C. Oxygen saturation levels. D. Respiratory rate. The correct answer is C. A pulse oximeter primarily measures oxygen saturation levels in the blood, which is crucial for monitoring respiratory and cardiovascular function. What parameter can be inferred from the reticulocyte count in a blood sample during physiological experiments? A. Platelet function. B. Bone marrow activity. C. Immune response efficiency. D. Hemoglobin levels. The correct answer is B. The reticulocyte count indicates bone marrow activity, providing insights into the production of red blood cells and overall hematopoiesis. How does a hemoglobin electrophoresis test assist in physiological studies related to blood disorders? A. To determine the clotting time of blood. B. To assess the iron levels in red blood cells. C. To identify abnormal hemoglobin variants. D. To measure the platelet aggregation rate. The correct answer is C. Hemoglobin electrophoresis helps in identifying abnormal hemoglobin variants, which is essential for diagnosing various blood disorders and genetic conditions. What information can be derived from a reticulocyte production index calculation during blood analysis in physiology experiments? A. Red blood cell turnover rate. B. White blood cell differential count. C. Platelet aggregation efficiency. D. Overall blood volume status. The correct answer is A. The reticulocyte production index calculation provides information about the red blood cell turnover rate, indicating the bone marrow's response to anemia or erythropoietic stress. Why is measuring mean arterial pressure crucial during blood pressure estimation in physiological studies? A. To assess diastolic function. B. To calculate cardiac output. C. To evaluate tissue perfusion pressure. D. To monitor baroreceptor activity. The correct answer is C. Measuring mean arterial pressure is crucial for evaluating tissue perfusion pressure, which reflects the blood flow to various organs and tissues in the body. What is the function of a hemocytometer in cell counting during physiological studies? A. To measure cell viability. B. To estimate total cell count. C. To assess cell morphology. D. To calculate cell size. The correct answer is B. A hemocytometer is used to estimate the total cell count by counting cells in a specified volume. What parameter can be assessed through a spirometry test in respiratory physiology studies? A. Heart rate. B. Blood pressure. C. Lung function. D. Liver function. The correct answer is C. Spirometry tests are used to measure lung function parameters such as vital capacity, forced expiratory volume, and more. What does a platelet count measurement in physiology experiments help in evaluating? A. Blood clotting ability. B. Red blood cell count. C. White blood cell count. D. Hemoglobin levels. The correct answer is A. Platelet count measurement helps assess the blood's ability to clot properly. What is the primary purpose of using a pulse oximeter in physiological studies involving oxygenation monitoring? A. To measure heart rate. B. To assess lung function. C. To determine blood pressure. D. To measure oxygen saturation in the blood. The correct answer is D. A pulse oximeter is used to measure the oxygen saturation levels in the blood. Why is monitoring blood pressure continuously important during physiological experiments? A. To assess red blood cell count. B. To monitor hemoglobin levels. C. To evaluate cardiovascular function. D. To measure lung capacity. The correct answer is C. Continuous blood pressure monitoring helps in assessing cardiovascular function throughout the experiment. What does the reticulocyte count indicate about bone marrow function in a blood sample during physiological experiments? A. Platelet production. B. Red blood cell production. C. White blood cell production. D. Lymphocyte production. The correct answer is B. A high reticulocyte count indicates increased red blood cell production, reflecting bone marrow function. What does a buffy coat layer in a blood sample indicate during physiological studies? A. Red blood cell count. B. Platelet count. C. White blood cell count. D. Hemoglobin levels. The correct answer is C. The buffy coat layer represents the white blood cell count in a blood sample. What is the significance of using a hemoglobin electrophoresis test in physiological studies related to blood disorders? A. To assess platelet function. B. To measure red blood cell count. C. To identify hemoglobin variants. D. To calculate mean corpuscular volume. The correct answer is C. Hemoglobin electrophoresis helps identify different hemoglobin variants present in blood, which is crucial for diagnosing various blood disorders. Why is cell counting important in physiological studies involving microscopy? A. To determine blood pressure levels. B. To monitor hemoglobin levels. C. To assess tissue structure. D. To quantify cell populations. The correct answer is D. Cell counting is essential in determining and quantifying cell populations when using microscopy in physiological studies. How does a centrifuge assist in blood analysis during physiology experiments? A. To determine cell morphology. B. To separate blood components. C. To measure hemoglobin levels. D. To assess enzyme activity. The correct answer is B. A centrifuge is used to separate blood components based on their density, aiding in various blood analyses during physiology experiments. What is the main benefit of heart rate monitoring during physiological studies? A. Assessing respiratory function. B. Determining blood glucose levels. C. Evaluating cardiovascular health. D. Measuring white blood cell count. The correct answer is C. Heart rate monitoring helps in evaluating cardiovascular health by providing insights into heart function and overall cardiovascular well-being. What is the purpose of cell counting in physiological studies? A. Assessing liver function. B. Evaluating kidney function. C. Estimating brain volume. D. Quantifying cell populations. The correct answer is D. Cell counting serves the purpose of quantifying cell populations, which is crucial for various physiological analyses and research. What can be inferred from a high white blood cell count in a blood analysis during a physiology experiment? A. Decreased immune response. B. Inflammation or infection. C. Low oxygen levels. D. Hypertension. The correct answer is B. A high white blood cell count often indicates inflammation or infection in the body, as it is a response to pathogens or foreign substances. What is the significance of platelet count measurement in physiology experiments? A. Assessing kidney function. B. Monitoring liver enzymes. C. Evaluating blood clotting ability. D. Measuring red blood cell count. The correct answer is C. Platelet count measurement is important for evaluating the blood's ability to clot properly, which is crucial for wound healing and preventing excessive bleeding. In physiology experiments, what is the main purpose of using a spectrophotometer? A. Analyzing cell structure. B. Measuring enzyme activity. C. Determining chromosome count. D. Estimating blood flow rate. The correct answer is B. A spectrophotometer is commonly used to measure enzyme activity in physiological studies, providing valuable information about biochemical reactions and processes. Which of the following devices is commonly used to determine the oxygen saturation in blood samples during physiological studies? A. Sphygmomanometer. B. Spirometer. C. Pulse oximeter. D. EKG monitor. The correct answer is C. A pulse oximeter is frequently utilized to measure the oxygen saturation levels in blood samples, offering insights into respiratory function and oxygen delivery in the body. What does a centrifuge help in achieving during blood analysis in physiology experiments? A. Separating blood components. B. Measuring blood pressure. C. Monitoring heart rate. D. Analyzing lung function. The correct answer is A. A centrifuge assists in separating blood components based on density, allowing researchers to analyze different blood constituents and study them individually. In physiology research, what is the primary purpose of using a hemocytometer for cell counting? A. Assessing hormonal levels. B. Quantifying cellular populations. C. Measuring bone density. D. Evaluating kidney function. The correct answer is B. The primary purpose of using a hemocytometer for cell counting is to quantify cellular populations accurately, aiding in various physiological analyses and research. Which of the following is a key advantage of using flow cytometry in cell analysis during physiology experiments? A. Measuring blood pressure. B. Quantifying cellular properties. C. Detecting enzyme activity. D. Evaluating liver function. The correct answer is B. One of the key advantages of using flow cytometry in cell analysis is its ability to quantify various cellular properties, such as cell size, granularity, and protein expression levels, with high accuracy and efficiency. What information can be derived from a differential white cell count in a blood analysis during physiology studies? A. Lung capacity assessment. B. White blood cell types ratio. C. Stem cell count. D. Heart rate variability. The correct answer is B. A differential white cell count provides information about the ratio of different types of white blood cells present in the blood sample, which is essential for assessing immune function and detecting potential health conditions.